Maya and Ronen, you talked about the interplay between cancer predisposition, the genomic variations in germline each of us has that make us more or less susceptible to developing cancer, and somatic mutations, the mutations that our cells accumulate during our lives. In this unit, I will try to highlight both of these aspects of cancer. I would like to start this segment by describing a once fatal hematologic disorder called chronic myeloid leukemia, or CML for short. This is a disease of enhanced proliferation of the blood cells, leading to a very high white blood cell count, as you can see in this picture. On the left, you see a few white blood cells in the peripheral blood having a pink-purple nucleus. This is a normal blood smear. On the right, a very high number of white cells are present, which is abnormal, and here, suggestive of leukemia. CML was the first malignant disease in which the genetic cause was found. In 1959, looking at cells during division, a unique chromosome was found only in CML cells and was called the Philadelphia chromosome. Only in 1973, using G-banding techniques, Dr. Janet Rowley had the seminal discovery that this was the result of a translocation between chromosomes 9 and 22. In this panel, you can see two sets of chromosomes. While the left chromosome 9 and the left chromosome 22 are normal, their partners look different due to this rearrangement, in which a part of chromosome 22 is fused to chromosome 9. Through this, there is uncontrolled activation of the ABL1 gene, and the diseased cells continuously signal and proliferate. This translocation resulted in a rearrangement of two genes, the BCR gene on chromosome 22 and the ABL1 gene on chromosome 9. This finding was not solely used as a diagnostic measure, but led to one of the first targeted therapies in leukemia. The BCR-ABL1 protein shown in this cartoon has an ATP binding pocket, which is required for its function. This is seen here as the ATP, in yellow, binds to the pocket with 3 phosphate atoms colored in green and uses them to phosphorylate the tyrosine residue of its substrate, which activates it. After understanding this vulnerability, a drug was designed to bind to that pocket and block it. This drug, imatinib, shown in the right side, binds to the same site but does not have the capacity to phosphorylate and thus activate the substrate, resulting in inactivation of the BCR-ABL1 protein. This breakthrough transformed CML from a deadly disease to a chronic condition that can be controlled by taking a daily pill. Moreover, this led to transforming the way we think of targeting cancer. For many years, we have known that a series of genetic mutations cause cancer transformation in a cell. Following the success of BCR-ABL1 targeting, researchers and clinicians from all over the world reasoned that if we identify vulnerable genetic mutations in malignant cells, we can find the right drug and stop it. In 2003, the Human Genome Project was completed. Since then, sequencing of entire human genome became more simple and cheaper, dropping from more than $1 billion initially to $1,000 per genome. This led to sequencing not only of normal genomes, but also of cancer genomes, in which we learn about the mutations occurring and defining each individual cancer. All these mutations are now cataloged in databases such as COSMIC and TCGA, which are described in the bioinformatics sessions. You will hear where to find these databases and how to use them in practice lessons. Again, the thought is that through understanding the mutated gene or disrupted pathway in the malignant cell, we can target the tumor specifically and improve the prognosis of individual patients based on their specific composition of the tumor DNA. An illustrative case demonstrating this strategy is of an infant born with a tumor in her tongue that obstructed her airway. This tumor was surgically resected, a very complex procedure in such a young baby. Despite this, it recurred almost immediately. This rare tumor is called hemangiopericytoma. According to standard pediatric protocols, chemotherapy was administered, but unfortunately with no success. Next, sequencing of the tumor was performed. Among several mutations which were detected, a mutation in the gene called BRAF was found. This mutation changes position number 600 in the protein, which is the most common mutated amino acid in other cancers like melanoma, but was never described in this rare hemangiopericytoma. Information about this mutation can be found in databases such as My Cancer Genome, which will be presented in the practice sessions. Importantly, this mutation can be targeted by inhibiting the active mutated BRAF using a drug called vemurafenib. Indeed, initiation of this drug led to disappearance and continuous control of this tumor, showing the importance of identifying the vulnerability of the tumor. Since every cancer is different and every patient is different, this process is difficult to study in the way standard clinical trials are designed, when taking into account hundreds and thousands of patients. Thus, individualized trials are to be designed, aiming to identify targetable genetic variants in cancer.